Hey guys, welcome to AP Biology. This is Coach K, and we're going to talk about the light reactions in photosynthesis part two. So, if you have your notes available, you should probably pull those out so you can kind of follow along. Okay, so this is a great little picture that kind of summarizes everything. We have the light reactions are called the light reactions because they require light, and light provides the energy to take water, break the water, separate water into electrons, protons, and oxygen, utilize those electrons, which will be energized by the light, in order to make ATP and to reduce NADP to NADPH. And the waste product of this whole thing will be oxygen, as you can see on the right hand side over here. Okay, so here's just another picture of it. I do want you to know the reactants and the products of all of these reactions. And as you can see, we have water coming in at the top. And process is occurring in the chloroplast and specifically on the thylakoid membrane, which is the inner membrane of the chloroplast. And this is where the chlorophyll pigments are located, and it is the chlorophyll pigments and other accessory pigments that will be absorbing the light energy, I should say, the photons, which consist of energy, and that energy is going to be used to boost electrons to a higher energy level. And that's going to allow the processes required in order to, create the energy needed in order to make ATP and also to reduce NADP. And as you can see, we have oxygen coming out of the chloroplast. Now, this is just oxygen is just a byproduct. It's of the breakdown of water. Okay, the plants don't really need the oxygen that much. Okay, so you may be wondering, how does a pigment capture light energy? Well, it does so by absorbing that energy in the specific atoms of chlorophyll. In the center of the chlorophyll molecule is a magnesium atom, and that has two valence electrons. Now, this is a very simplified picture of electron energy shells, okay, or orbits. It is a model. It is not accurate in real life. However, models allow us to simplify what's going on so we can actually understand it. When you get, or have, if you've already taken chemistry, you know that there's p orbitals, s orbitals, z orbitals, and they have lots of different shapes. But this is a simple Bohr model, BOHR, which does allow us to basically look at what's actually going on. So these are, we see three energy level shells surrounding this atomic nucleus. And all I'm going to say is that the closer the electrons are to the nucleus, the less energy they have. So when the magnesium atom at the center of the chlorophyll molecule absorbs photons' energy, those electrons are going to go to a higher energy level. So if they're sitting at the first energy level, they'll go up to the third. And when they are at that high energy level, they're very unstable. They don't want to hang around the magnesium very much. And so they want to actually escape. And that's what's going to sort of happen. All right. So when we talk about the light reactions, okay, they are divided up into three primary stages. We have the primary photo event, which I'll discuss shortly. We have electron transport, and then we have chemiosmosis. All right. So first of all, we have to have the primary photo event. That's when photons strike chlorophyll molecules on the thylakoid membrane and excite electrons that are around the magnesium. Well, this causes the energy to be transferred from one chlorophyll molecule to an adjacent chlorophyll molecule, and it kind of bounces around, almost like if you are playing pool. The first thing you need to do is break the rack of balls, and you do that by taking your pool stick and then hitting the cue ball. And when the cue ball hits that rack of balls, oftentimes the cue ball just stops. Well, what happens to the energy? The energy is transferred to all the other balls, and they go scattering around the table. Kind of like what happens here. Photons hit these these chlorophyll molecules, the magnesium or the electrons don't really move, but the energy is transferred. And the energy keeps on bouncing around until it gets to a special chlorophyll molecule called the reaction center chlorophyll. And then electrons absorb all that energy and they get boosted up to a higher energy shell where another adjacent protein is going to grab those electrons at the beginning of what's called the electron transport chain. So here is our Z diagram. This is in your textbook, and this is a diagram of non-cyclic electron flow. I just want to say that all of this, what you are looking at on this particular picture, are all embedded in the thylakoid membrane, which makes up the inner membrane, that highly folded inner membrane inside of the chloroplast. So this membrane right here where you see the light reactions. So again, the photosystems are clusters of chlorophyll molecules. And so light energy is going to come in. It's going to basically bounce around, excite a bunch of these electrons until 2 electrons get boosted up to a higher energy level. An adjacent protein is going to grab those 2 electrons and pass it down this sequence of proteins first from the primary acceptor to PQ, which stands for plastoquinone, which is a protein that can actually move within the membrane. And it's going to pass those electrons to a cytochrome complex. The cytochrome complex is actually a proton pump. And it's going to use the energy in the electrons to pump protons from a low concentration to a high concentration. Since it's using energy and it's going from low to high, this is a form of active transport that does not use ATP. Instead, it uses the energy of these electrons. I also want you to notice on the left-hand side of this picture, there's this arrow that shows the energy of electrons. So down at the bottom, we have low energy electrons, and that's what you see in these photosystems. And as sunlight comes in, it boosts the energy of the electrons much higher. And so up here at the primary acceptor, the electrons have a lot of energy. And as they move down the electron transport chain, they lose energy. Why? Because they're using the energy to pump protons against the concentration gradient. And then once you have that concentration gradient, we're going to use that to make ATP, as you see here on the bottom. Okay, so that's product 1 of the light-dependent reactions. So by the time we get to the end of the electron transport chain, the electrons have now been depleted of energy, and they now go into photosystem 1, where once again more light is coming in, boosts them up to a higher energy level, where once again they go down a very short electron transport chain, and instead of being used to pump protons, these 2 high energy electrons are going to be used to reduce NADP to NADPH. This is an electron carrier or electron transport molecule that is carrying high energy electrons that are going to be used to reduce carbon dioxide in the next stage. Remember, oil rig, oxidation is loss and reduction is gain. Besides non-cyclic electron flow, there's also cyclic electron flow. And let me just show you why is this called non-cyclic electron flow. It's because the electrons start here in photosystem 2, and then they're going to go down the electron transport chain, go down another electron transport chain, and then they'll end up here on this molecule of NADPH, and then the NADPH will be used to make sugars. And so the electrons don't come back. They're going one way. That's why it's called non-cyclic electron flow. Cyclic electron flow, on the other hand, basically goes in a little circular cycle, and it only involves photosystem 1, not photosystem 2. And you may also be wondering, why is photosystem 2 called photosystem 2 and photosystem 1 when the first event happens in photosystem 2? Well, long and short story of that is that the photosystem 1 was discovered first, and that's it. Okay, so in cyclic electron flow, all that happens is that electrons get excited, they go to a short electron transport chain, they pump protons against the gradient, and that gradient is going to be used to make ATP, and the only product we see with cyclic electron flow is going to be ATP, not NADPH. And this is what many bacteria do, Plants, on the other hand, and other green, more complex organisms besides bacteria, autotrophs, they do both because in the production of sugars, more ATP is needed than NADPH. So plants do both of these. A lot of bacteria only do cyclic electron flow. So as I said earlier, these photosystems and the electron transport chains are all located within the inner thylakoid membrane. The more thylakoid membrane you have, the more electron transport chains you have, the more photosystems you have, so on and so on. But let's look at this diagram. This is a really good diagram, a more realistic one. We have photosystem 2 right here and photosystem 1, and they are connected by the electron transport chain. Again, the light energizes electrons in photosystem 2. They're passed to a high energy, to an electron transport carrier, plastoquinone. Those electrons are then brought to the cytochrome complex, which is going to use the energy of the electrons to pump protons from a low concentration within the stroma to a high concentration in the thylakoid space. Now, there's about a 1,000-fold difference of proton concentration between the stroma and the thylakoid space. That's a huge concentration gradient. And it's only made possible because of this compartmentalization that we see in the chloroplast and in the mitochondria, where different areas are separated by a phospholipid bilayer that is going to prevent the movement of electrons simply via diffusion through the membrane. Because they have a charge, they cannot go through that hydrophobic interior. Okay, so we see the cytochrome complex is helping to create that proton gradient. And then those electrons, which have now lost their energy, go to photosystem 1. They go down a short electron chain to NADP reductase. And then we're going to use those electrons to reduce NADP to NADPH. I do want you to notice that the two products, NADPH and ATP, are both going to be made within the stroma, which is where the Calvin cycle occurs. Okay, so we'll go on a little bit further. I do want to show you this picture right here. It's just a little cartoon, but all it's showing is that the more thylakoids we have, the more folds we have, the more proteins we have, the more pigments we have, the more ATP synthase we have, the more efficient this cell, this organelle has become. And do remember, the chloroplast is the result of endosymbiosis of once free-living prokaryotic organisms that were roughly circular in shape. And over evolutionary time of over 2 billion years, because that's when photosynthesis, according to the data that we have, pretty much came about or evolved, that engulfed bacterium has become highly folded into the thylakoid membrane that we do see today. And again, more surface area equals more energy, and that's the big picture right there. So you may ask, where does the water come in the process of photosynthesis? And as we discussed earlier, this is non-cyclic electron flow, so that causes a problem because once the electrons are boosted to a higher energy level and they start going down the electron transport chain, we now have a hole in this reaction center chlorophyll that is missing 2 electrons. We need to fill that hole with 2 electrons. Where do those electrons come from? They come from water. And light energy is going to split the water into 2 protons, 2 electrons, and half of an oxygen molecule. So it takes 2 water molecules to make an O2 molecule. And this is where these low energy electrons are coming from in order to replace the ones that get boosted up. And of course, they're low energy here, but once they get into that chlorophyll molecule and receive the energy from the other chlorophyll molecules, they get boosted up to a higher energy state, and on and so on and so on. So that is the role of water in photosynthesis, is to supply electrons, and a byproduct is oxygen. Okay? And again, we already discussed that. So down on the thylakoid membrane, the first thing that's going to happen is light energy is going to come in, and that light energy is going to be absorbed by chlorophyll pigments. It's going to pass that energy around until we get to the reaction center chlorophyll, which is going to boost 2 electrons up to a primary acceptor. Okay, in order to fill that hole that now exists, water is split to provide the 2 electrons necessary to fill the hole. And they'll get boosted up and so on and so on. Now, those high energy electrons are going to be used to pump protons against a concentration gradient. And we're going to use that concentration gradient in the last step, which is called chemiosmosis, in order to make ATP. Those low energy electrons now, no, I should say those electrons which have been depleted of their energy, now go to photosystem 2. Once again, light is going to energize those electrons. They go up to a primary electron acceptor, and then down another short electron transport chain where these 2 electrons are going to be used to reduce NADP to NADPH. And notice this, there's an enzyme here, NADP reductase. Oil rig, oxidation is loss, reduction is gain. And there's just a picture of how cyclic electron flow would work in order to make just ATP and not NADPH. So all of these are what we call redox reactions, okay? Here we have compound A, which has 2 electrons. It is a reducing agent. And when it gives those electrons, when it loses those electrons, compound A now becomes oxidized. And it's called a reducing agent because it's going to reduce compound B. And compound B gains those electrons. So the electrons go from A to B. And B is an oxidizing agent because it's able to steal those electrons or take those electrons, and it now becomes reduced as it gains those electrons. So just some terminology right there. So a couple of questions here. Is there any oxidation happening, or is anything being stripped of electrons? And you should say, yes, of course, water is. All right. Is there any reduction happening? Yes, of course. It's over NADP is being reduced to NADPH. Is there any catabolism happening? Anything being broken down? Yes, of course. That is going to be water being broken down. Is there any anabolism happening? Is anything being built? Yes, of course. That's going to be ATP. Okay. And is there any energy coupling occurring? Yes, we are using the breakdown of water to provide energy or provide the electrons to basically make ATP. And we're also doing some, so we got that going on. So let's look again at this right here, this particular diagram. As I said earlier, there is a 1,000-fold decrease, or I should say 1,000-fold increase of protons between the stroma and the thylakoid space. This is a pH of about 7, and this is a pH of about 4, if I remember correctly. So it's about a 1,000-fold difference, okay? So there are 3 places where this concentration gradient is created. Number 1, the splitting of water. It occurs inside the thylakoid space, and so that's going to increase the proton concentration. Number 2, the electron transport chain. Okay, that is going to pump protons from a low concentration in the stroma to a high concentration in the thylakoid space using the energy of these high energy electrons. So our third place that's going to our third area where that's going to contribute to the proton gradient is right here at NADP reductase. And NADP reductase is going to reduce the amount of protons in the stroma, I should say the amount of free protons in the stroma, which will then also aid to increase the proton concentration gradient. So what do we do now? We now have lots of protons inside the thylakoid space, 1,000 times more. Well, these protons can now diffuse through ATP synthase, which is a protein. What type of transport is that called? If you said facilitated diffusion, you are absolutely 100% correct, because we're going from a high concentration to a low concentration via a specialized transport protein. And this specialized transport protein is going to basically take ADP, adenosine diphosphate, plus a free inorganic phosphate and create ATP. Once again, both of these processes, reduction of NADP and production of ATP, are going to occur in the stroma, which is where the Calvin cycle is. Okay. And again, cyclic electron flow is used because in order to make sugars in the next stage, the light independent reaction or the Calvin cycle, more ATP is needed than is NADPH. So we generally think of what makes ATP in a cell. Most people go, it's the mitochondria, the powerhouse of the cell. Well, the chloroplast can also make ATP. However, it's different because instead of using energy derived from electrons from macromolecules, it's using low energy electrons from water that get energized by light. Okay. And all of the probably all of the ATP that is generated during, or I should say inside the chloroplast, is utilized to make sugars. And then those sugars will be broken down by the plant in the mitochondria to make ATP available for doing cellular work. Okay. Cytochrome C is an electron pump, or excuse me, a proton pump, okay, that is powered by electrons. And every single organism that we've ever looked at has this, from bacteria to blue whales to you and I. And this cytochrome complex is it's a protein, so therefore it's made up of amino acids, and the sequence of those amino acids is encoded in your DNA or in every organism's DNA, and it's what we also call a evolutionarily conservative molecule, meaning that it changes very slowly over time. And so molecular archaeologists have utilized cytochrome C changes, the changes in both the amino acid sequence or even in the nucleotide sequence of the gene, to look at relatedness between different species of organisms. The more similar they are, the more recent they've had a common ancestor. I think, if I can remember correctly, the cytochrome C complex between us and chimps, there are no differences at all. That divergence occurred about 7 million years ago, which to you and I seems like a long time, but in evolutionary terms, it's all but a blink in the eye. Now, we can use cytochrome C differences to compare differences between, or times of divergence between, much more distantly related species, let's say humans and mice, humans and bugs, humans and fish, humans and plants, things like that. And we can start getting some good data from that. And again, that concentration gradient is a form of potential energy, just like water behind a dam. Remember, 1,000 times more inside that thylakoid space than there is in stroma. When the protons diffuse down through this molecule, this enzyme called ATP synthase, which consists of two rotors around a shaft, this rotor is going to spin. The potential energy of the proton gradient is going to be turned into kinetic energy as the protons move through that, which is going to spin this, that's a form of mechanical energy. And then the bottom part, we're going to take ADP plus a phosphate and put them, and that spinning rotor is going to put those two together to make ATP, which is a form of chemical energy. So we go from potential energy to kinetic energy to mechanical energy back into chemical energy. Whew, that's a lot of energy transformations. All right, and we'll look at a couple of, you can Google, ATP synthase animation to get some views of what this molecule, this enzyme looks like. It's pretty fascinating. Okay. So again, what do the light reactions produce? They produce energy in the form of ATP, and they don't produce electrons, but they create, or not even create, they harvest electrons is a better word to use, from water and protons, and they use those to reduce carbon dioxide to sugar molecules, CH2O, in the light-dependent reaction. Okay, so again, all we're doing is converting light energy into chemical energy. All right, so as I say here, electron transport chains are redox reactions that transfer electrons from an electron donor to an electron acceptor. The transfer of electrons is coupled to the movement of protons across a membrane, producing a proton gradient. And then this proton gradient is used to produce useful work. And that is it, guys. I hope you enjoy.